In this video, I'm going to teach you more about the details of the cell-mediated response. The cell-mediated response involves T cells, specifically cytotoxic T cells, whose job are to locate infected cells or abnormal cells, such as cancer cells or transplanted cells, and lyse them. Now, lysing these abnormal infected cells would ideally kill cells that might be harmful because they are allowing viruses to reproduce or are growing abnormally, like with cancer, or just don't belong in the body, like something like a transplant might be. Now, cytotoxic T cells know which cells to attack because of MHC1 molecules. We learned about these in a previous video. MHC1 molecules are found on body cells, and they present specific antigens. Cytotoxic T cells that are specific for that antigen will bind to those class 1 MHC molecules, release chemicals called perforin that lyse those cells, destroying the cell and stopping the potential infection. Now, we are going to learn a little bit more about what it means to be an infected cell first, and then we'll walk through the pathway of a cytotoxic T cell. So either being directly stimulated, as we see here, or being stimulated by a helper T cell. So we'll review both of those. All right, so when we talk about infected cells, we're talking about viruses. So we need to learn a little bit more about viruses to understand how exactly they might have antigens that could be presented on an MHC molecule. So viruses in general, they have some basic components. They have genetic material. It could be DNA or RNA. And because RNA is the genetic material, that's one of the reasons why viruses aren't considered living things. They also have something called a capsid, which is this protein coat. Now, viruses come in lots of different shapes and sizes, but they have those two basic components. So here you can see there's kind of a tubular capsid, and then this one has RNA inside. This one right here looks kind of like an adenovirus. It's got this kind of protein, almost geometric outside, and then it has DNA on the inside. Some viruses have a membrane around them. They have a membrane envelope that they pick up from the cells that they infect. And this one right here, which we will be talking a lot about because a lot of experiments are done with this thing that looks kind of like a moon lander is called a bacteriophage. So this is a virus that specifically attacks bacteria cells. So viruses, protein coat, some sort of genetic material on the inside. Now, how exactly do viruses infect cells? Well, we're going to be looking at this example that involves phages, bacteriophages, which are special viruses that attack bacteria. But the same premise could apply to a virus attacking a human cell in the body. So we're going to do a phage and we're going to do a bacteria cell. Now, viruses infect cells by injecting their genetic material into the cell. So here you can see this phage DNA is being injected into this bacteria cell. Once the genetic material is in the cell, there are kind of two possibilities. There's the lytic cycle and the lysogenic cycle. Let's start off with the lytic cycle. So once the genetic material is inside the host cell, it can then be read and used to make more proteins and DNA. So basically, think about an infected cell kind of as a virus-making factory. The virus releases chemicals and activates genes that basically take over cell functions, and the cell uses all of its machinery, its ribosomes, its enzymes, to construct viral proteins and viral genetic material. Those get assembled, new viruses are made, and then those viruses oftentimes break out of the cell. So the cell lyses to release the virus so it can go infect other cells. It's what we call the lytic cycle. So it's like the reproductive cycle, okay? The host cell becomes a virus-making factory, the viruses get assembled, and they break out of the cell. But some viruses go through this period of kind of dormancy. And what can happen is that viral DNA, that blue circle right there, sometimes it gets incorporated into the host cell's chromosome, or host cell genome, we like to call it. So in this case, because it's a phage DNA, it's called a prophage. In our cells, for example, HIV will incorporate, once the RNA has been copied to DNA, that viral DNA gets incorporated into our genomes, and we call that a provirus. So when you hear that pro and then some sort of virus or phage, that is DNA that is incorporated into the host cell's genome, so viral DNA. So basically, now we have this incorporated DNA, and it can hide inside the cell. And as that cell reproduces, it copies and passes on that viral DNA to new cells. So it's another way to kind of keep passing on the virus. But notice, it's not making any new viruses, it's just kind of hiding within the cell. So here you can see, here's a daughter bacteria cell with that prophage DNA. Now, occasionally, something's going to happen that triggers that DNA to leave the chromosome, and it pops out. So in humans, oftentimes, this includes things like stress. When you have released stress hormones, often your immune system gets suppressed, and so as a result, increased stress means there's less to fight off those viruses, and so it can kind of get established in your body, or it could be a weakened immune system due to age or due to some other illness. Now, this phage breaks out, and now it can go into that lytic reproductive cycle and make new viruses. So sometimes you can be infected with a virus and have no symptoms because the virus is being dormant inside your cells. All right, so that is how viruses infect cells. So let's say we have a body cell that's infected. How does it alert cytotoxic T cells to this infection? So this is the part we need to focus on. This is the cell-mediated response. TC cells, those are cytotoxic T cells, also called killer T cells, also called CD8 cells. So these cytotoxic T cells get activated by binding directly to an antigen-presenting cell. Sorry, I forgot to write that word in. So here you can see, here's our cytotoxic T cell. Okay, it is using receptors on its surface to bind to an infected cell. So here are these foreign antigens. So when the virus is reproducing inside that host cell, some of those viral proteins can get incorporated into class 1 MHCs on their surface. So the MHC is presenting this antigen to the cytotoxic T cell. That cytotoxic T cell is specific for that antigen. Once the binding has occurred, the cytotoxic T cell releases a chemical called perforin. Perforin perforates the infected cell. It makes these holes, and you can see that the cytoplasm leaves through those holes and the cell lyses. Dead cell, virus can't reproduce. Virus depends on its host cell for its reproductive capabilities. So that is one way that cytotoxic T cells are activated. They can be activated directly by an antigen-presenting infected cell. Now, I keep saying infected cell because we just talked about viruses infected cell, but this could also be a cancer cell that's presenting an abnormal protein. So cancer cells have many mutations, typically 6 to 10 mutations in genes that regulate the cell cycle are mutated, and as a result, they can produce abnormal proteins that show up on their surface. The immune system cells, those cytotoxic T cells, recognize these abnormal proteins, and they release perforin and lyse those cancer cells. Transplants, instead of an infected cell, let's say it's a transplanted cell, that transplanted cell is going to have MHC markers that the body doesn't recognize as belonging to itself. Okay, that transplanted organ came from another person who has these different MHC molecules due to the genes they possess. As a result, the immune system cells, specifically those cytotoxic T cells, are going to bind to the MHCs on the transplanted organ, release perforin, and lyse it. Okay, so that's one way cytotoxic T cells can be activated. directly by an antigen-presenting cell. The other way that T cells can be activated in the cell-mediated response is actually a faster way, which involves helper T cells. So if you remember back to the previous video, macrophages engulf some sort of foreign antigen, so in this case a virus. They present antigens on their surface with class 2 MHC molecules. Okay. A helper T cell would then bind to those antigens on the MHC molecules, become activated, and release chemicals that basically recruit cytotoxic T cells and activate them. Once the cytotoxic T cell becomes activated, it then goes through the process of binding to infected cells and releasing perforin and then destroying those foreign cells. So this process where the helper T cell is actually recruiting cytotoxic T cell, because the macrophage in fact recruited those helper T cells, that's actually a pretty fast process. The cell knows exactly what it's looking for. When the immune system is relying on direct binding to antigen-presenting cells, that's going to be slower because by chance, the right cytotoxic T cell has to come in contact with the right infected cell. So it can take a little longer versus the recruiting process involving macrophages and helper T cells, which is more targeted and kind of, because of chemical communication, recruits more cells faster. So I hope this helps you understand how cytotoxic T cells work and how exactly cells can get infected.